Imagine a future where the code of life itself, DNA isn't just something we read, it's something we can actively edit,
Uh,
where we could maybe correct genetic diseases before they even show up, or engineer crops to handle, well, pretty much any climate change throws at them.
right.
Or even, and this is wild, glimpse the possibility of bringing back species that are extinct.
That sounds like science fiction, doesn't it?
It really does, but it's not. It's the remarkable and sometimes, frankly, a bit daunting reality of CRISPR gene editing.
Mm-hmm.
This tool is just so precise, you can really think of it like molecular scissors for DNA, making incredibly specific changes.
Tiny little cuts, exactly where you want them.
So today we're diving deep into CRISPR. We want to unpack its incredible origins, which are actually quite surprising.
They really are. Not what you'd expect.
And its transformative applications, across medicine, agriculture, and of course the really big ethical questions it raises
Yeah.
about our role in, well, rewriting life's blueprint.
Yeah, the heavy stuff.
We'll explore the science, the promises, the perils too, and what it all means for our shared future.
And what's just fascinating here is that we're talking about a technology that it really emerged from one of the most ancient battles on Earth. I
Right.
mean, microscopic scale, but still.
Which battle is that?
The constant war between bacteria and viruses. Phages attacking bacteria. Understanding that fundamental biological process was the key. Completely unexpected.
Wow.
It unlocked what's become arguably the most versatile genomic engineering tool we've ever had.
So basic research into bacteria fighting viruses led to this.
Exactly. It's a real testament to how, just deeply observant science, fundamental research, can spin off into these groundbreaking applications.
That is fascinating. Bacteria having this kind of built-in memory system against viruses. It makes you wonder, right? If nature developed something so sophisticated, what else is out there? What did that teach us about biological ingenuity?
Well, it certainly showed us that evolution comes up with some incredibly elegant solutions, things we might not have appreciated before.
Okay, so let's unpack this. We're talking about CRISPR, clustered, regularly interspaced short palindromic repeats.
Quite a mouthful. Yeah.
It is. But these really strange repetitive DNA sequences, they were first spotted in bacterial DNA way back in the 1980s.
That's long ago.
Yeah. And scientists were just completely puzzled for years. They looked like, I don't know, mysterious bookmarks in the genome, repeating bookmarks, no obvious function.
Just sitting there.
What were they? What did they do? Nobody really knew. It was a genuine biological mystery hiding in plain sight.
Exactly. For the longest time, they were just an oddity, a curiosity in the bacterial genome that didn't really fit anywhere. Researchers would note them, sure, but their actual purpose, it was elusive until around 2005. That's when a significant breakthrough started to unravel it. Researchers figured out these CRISPR sequences were actually part of, well, an ancient, pretty sophisticated immune system.
An immune system in bacteria.
Yeah, used by bacteria.
How does it work?
So when a virus attacks a bacterium, the bacterium does something remarkable. It captures a tiny piece of the virus's DNA, snips it out,
Okay.
and then it stores this snippet, almost like a mugshot, you could say, right there in its own genome, within those CRISPR repeat sequences.
So it keeps a record.
Exactly. It acts as a memory. So if the same virus attacks again, the bacterium can quickly recognize it using that stored snippet and, well, destroy it.
That's incredible.
It's nature's own adaptive immunity, but on a microbial scale. A bit like how our own immune system develops antibodies after an infection, right? It learns.
The elegance of that natural system is, wow, truly remarkable. Like bacteria develop their own genetic most wanted posters for viruses.
That's a great analogy, actually.
But here's where it gets really transformative, isn't it? This natural defense mechanism was just sitting there waiting to be, well, repurposed by us.
Precisely.
And that pivotal moment came in 2012. Two brilliant scientists, Jennifer Doudna and Emmanuelle Charpentier, they realized this bacterial defense system could be turned into a programmable gene editing tool.
That was the leap, the insight.
Their groundbreaking work, published in Science that summer, it didn't just win them the Nobel Prize in Chemistry in 2020 for, rewriting the code of life.
Which was huge, obviously.
Absolutely.
Yeah.
It fundamentally changed our understanding of what's possible in biology, just turned everything on its head.
It was a truly revolutionary insight. They figured out the core components that make this bacterial system work, and crucially, how to simplify them and harness them.
What are the core components?
Okay, so first there's the Cas9 protein. You can absolutely think of this as the molecular scissors. It's an enzyme, right? A biological catalyst, and it makes a precise double-stranded cut in DNA, but only at a very specific site.
Okay, the scissors. Got it. What else?
Then there's the synthetic guide RNA, or gRNA. This is a really clever bit. This provides the precision, the programmability. Well, the gRNA is meticulously designed. It has two parts. One is a guide sequence. That's a short stretch of RNA letters, nucleotides, that matches, it's complementary to the target DNA sequence you actually want to edit.
So it finds the right spot on the DNA?
Exactly. And the second part is a scaffold sequence. This bit binds to the Cas9 protein, essentially holding the scissors in place and telling them where to cut based on where the guide sequence is landed.
So if I'm getting this right, the guide RNA is basically the GPS navigating the system.
Perfect analogy.
And the Cas9 protein is the, I don't know, the guided missile or the scissors that arrive at the destination.
Yeah, the scissors probably best. It directs the Cas9 exactly to the spot you want in the DNA, and then Cas9 makes its cut.
And the truly incredible thing is scientists can target, virtually any gene in any organism just by changing that little guide RNA sequence.
That's the power of it. You just swap out that 20 nucleotide guide sequence to match a different target gene, and the whole system redirects itself.
It's like having a universal molecular search and destroy tool, or maybe search and replace sometimes. And it's customizable just by changing a few genetic letters.
That analogy really captures the simplicity and the power, and its incredible versatility. But, when you say virtually any gene, there are still some practical limits, right? Current challenges. Are some genes harder to target?
That's a good point. It sounds almost too easy.
Well, the principle is straightforward, but yeah, real-world application always has nuances. Delivery into the right cells can be tricky. Efficiency isn't always 100%. Potential off-target effects need careful checking, but the core concept holds.
And the original team, Doudna and Charpentier, they must have done rigorous checks to prove this worked.
Oh, absolutely. Incredibly rigorous steps to validate the system. They started with in vitro experiments.
Yeah.
That means in test tubes, right? Using purified components. Yeah, Cas9, gRNA, and target DNA.
To prove the basic mechanism.
Exactly. To confirm that the gRNA could effectively direct Cas9 and make those precise cuts on isolated DNA. That confirmed the fundamental mechanics. Then, once they had that nailed down in vitro, they moved to in vivo testing. So introducing the CRISPR-Cas9 system into living cells, bacterial cells first, then plant and animal cells.
Yeah.
And they meticulously monitor the DNA edits happening inside those cells.
That step from the test tube to a living cell, that seems huge. But how do you even check if the edit worked? I mean, at that tiny scale. And how do you know it only changed what you wanted it to change?
Excellent question. You need incredibly precise molecular biology methods. The gold standard is DNA sequencing.
Reading the DNA letters.
Pretty much. You read every single letter of the edited DNA region to make absolutely sure it's exactly right and only the intended changes were made. No unexpected mistakes nearby, no cuts somewhere else entirely.
Okay, what else?
Well, you also use PCR, polymerase chain reaction. Think of it like a molecular copy machine.
Right, makes lots of copies of a specific bit of DNA.
Exactly. It makes millions of copies of the targeted DNA fragment from the edited cells. That gives you enough material to analyze properly.
And then you analyze those copies.
You can, for example, run them on a gel electrophoresis. It sorts DNA fragments by size. This can quickly tell you if a cut was made where you expected, because sometimes the repair process introduces small insertions or deletions, changing the size.
Ah, okay. So sequencing confirms the exact change, PCR gives you enough material, and gels can give a quick check.
That's a good summary. These are the standard proof methods in molecular biology for verifying genetic changes. And while they confirm the immediate edit, predicting all the long-term biological consequences, especially in a whole complex organism, that's still an active area of research. Gene interactions are complicated.
It really is a testament to meticulous scientific work, though, taking it from this obscure bacterial defense thing to a tool we can actually program.
Absolutely.
But CRISPR, as powerful as Cas9 is, it isn't just that one static tool, is it? You mentioned it's more like a diverse family of systems.
That's right. We initially focused on Cas9, especially the one from Streptococcus pyogenes, SpCas9, because it's just really efficient and works on a wide range of DNA targets. It was the workhorse early on.
But once scientists understood how it worked,
the very next question was, okay, what else can we make it do? How can we refine this, tweak it? And that just led to this explosion of innovation. It really turned CRISPR from just scissors into an entire molecular toolkit.
A toolkit. I like that. So what else is in this toolkit?
Well, first, just to give some context, naturally occurring CRISPR systems are actually classified into different types.
Right.
Class 1 systems involve multiple proteins working together in a complex. Class 2 systems, like the one Cas9 belongs to, are simpler. They rely on a single protein, like Cas9 itself, to do the main job.
And that simplicity made them easier to engineer.
Much easier to repurpose, yeah. Now, the standard Cas9, the wild type, its power comes from making those double-stranded breaks in the DNA. Cut both strands.
Right, the scissors action.
And when the DNA is broken like that, the cell panics a bit and tries to patch it up. There are two main ways it does this. One is called non-homologous end joining, NHEJ. It's basically the cell just quickly gluing the broken ends back together. It's fast, but it's often messy. It can introduce small errors like adding or deleting a few DNA letters right at the cut site.
Which can be useful if you want to. What? Break a gene?
Exactly. It's great for knocking out a gene, disabling its function. But it's not precise if you want to correct a gene.
So what's the other repair pathway? The precise one?
That's called homology-directed repair, or HDR. Here, if you provide the cell with a template, a separate piece of DNA that has the correct, healthy sequence you want, the cell can use that template as a guide.
Like a blueprint for the repair.
Precisely. It uses the template to accurately copy and paste the correct sequence into the cut site. So instead of just sticking the ends together crudely, it repairs the break perfectly, fixing the typo with the right information. That's how you make precise corrections or insertions.
Okay, so Cas9 cuts. And then the cell either makes a rough patch, an HEJ, or uses a template for a perfect fix, HDR. But the innovation didn't stop there, right? You said scientists modified Cas9 itself.
They did. They started tinkering with the Cas9 protein. For example, they created something called nickase Cas9, or nCas9.
Nickase, like it just nicks the DNA.
Exactly. A single-point mutation, changing just one amino acid in the Cas9 protein, makes it so it only cuts one strand of the DNA double helix, not both. Just makes a nick.
Why would you want that? Isn't a double-strand break the whole point?
Well, double-strand breaks can sometimes be toxic to cells, and they increase the risk of unwanted larger mutations or rearrangements. A single-strand nick is much gentler.
So safer.
Potentially, yes. And it enables a technique called tandem targeting. You use two different guide RNAs to bring two nCas9 molecules to nearby locations, one nicking the top strand, the other nicking the bottom strand, slightly offset.
Ah, so you still get a break across both strands eventually, but it's staggered.
Exactly, a staggered double-strand break. This is generally less toxic, and it significantly improves specificity. It makes it much less likely that you'll get cuts at unintended off-target sites in the genome. It's about increasing precision and safety.
Okay, that makes sense. More control. And then there's dead Cas9. Cas9, that sounds, well, completely useless if it's dead.
It does sound counterintuitive. But dead here just means catalytically inactive. In Cas9, mutations in both of the cutting domains render the protein entirely unable to cut DNA. It cannot cleave DNA at all.
So, it does nothing.
Not quite. Crucially, it can still be guided to a target DNA sequence by the gRNA, just like regular Cas9. It binds to the specific address you send it to.
But it doesn't cut when it gets there.
Correct. And that makes Cas9 an incredibly versatile platform, because since it goes to a specific spot but doesn't cut, you can attach other molecular tools, other effector proteins to it.
Like hitchhikers.
Exactly. You use Cas9 as a delivery truck to bring those effector proteins to very specific locations on the DNA within living cells.
Whoa. Okay, so it's not just about cutting anymore. It's about targeted delivery of other functions. That feels like a really big shift.
It is. It fundamentally changes how we can interact with the genome. We're moving beyond just fixing broken genes or cutting them to actively controlling biological processes in a targeted way.
Could this delivery approach, using Cas9, actually be more useful than cutting and pasting sometimes, or is it just complementary?
It's often complementary, but in some situations, it's definitely superior, especially because it avoids the risks associated with making double-stranded breaks altogether.
And this led to, what, new kinds of tools?
Yes, it paved the way for what we call second-generation tools. Prime examples are the base editors.
Base editor. Okay, what do they do?
These are revolutionary. They typically use nCas9, the nickase, or Cas9, as the delivery platform. Attached to the Cas9 is another enzyme, usually a deaminase like a cytidine deaminase or an adenine deaminase.
Deaminase.
Yep.
It's like it changes an a base.
Exactly. These enzymes can chemically convert one DNA base directly into another. For example, acididine deaminase can change a C cytosine into a U uracil, which the cell then reads as a T thymine. And adenine deaminase can change an A adenine into an inosine, which the cell reads as a G guanine.
Without cutting both strands of the DNA.
Precisely. No double-stranded break required. Maybe a nick from NCAS9 to stimulate repair, but not the full cut. This allows for truly precise single-letter edits in the DNA code.
Wow. So it's like finding a single typo in a giant book and using, I don't know, molecular whiteout and a pen to fix just that one letter instead of tearing the page.
That's a fantastic analogy. It's much more subtle than the cut and repair mechanism of standard Cas9.
Yeah.
And we've seen this used, for instance, to introduce stop codons into genes.
Stop codons. Those tell the cell to stop making a protein, right?
Correct. So you can use base editing to effectively silence a gene by inserting an early stop signal, preventing the full protein from being made. Incredibly useful for research and potentially for treating diseases caused by faulty proteins.
So Cas9 isn't just for base editing. What else can this delivery truck platform do?
Oh, its applications are rapidly expanding. You can use it for epigenetic editing.
Epigenetic.
So
That's not changing the DNA sequence itself, but how genes are used.
Exactly. It's about altering the marks on or around DNA that control gene activity, turning genes on or off without changing the underlying A's, T's, C's, and G's. Think of it like changing the formatting in a document, bolding, italics, highlighting, without changing the words themselves.
Why is that useful?
Because many complex diseases involve problems with gene regulation, not just faulty gene sequences. So epigenetic editing opens up entirely new therapeutic strategies. DDCas9 can deliver enzymes that add or remove these epigenetic marks at specific genes.
Okay, what else?
It can be used for very precise gene regulation, fusing DCas9 to proteins that activate or repress gene expression. Lets you essentially install a dimmer switch or an on-off switch on specific genes. You can turn single genes or even multiple genes on or off at will. For chromatin imaging, you can attach a fluorescent tag, like GFP, to DCas9. Then you guide it to a specific DNA region. Now you can actually see that part of the chromosome inside a living cell nucleus. You can watch how DNA moves and organizes itself in real time.
You can watch genes moving. Seriously.
Seriously. And even more advanced, researchers are using it for manipulation of chromatin topology. That means altering the 3D folding and structure of DNA within the nucleus. Since structure affects function, this can profoundly impact gene activity.
It's like we're getting controls for the entire operating system of the cell, not just editing the code.
That's a great way to think about it. Our ability to interact with the genome is becoming incredibly nuanced.
And just when my brain is full with DNA editing, there's more. You mentioned RNA targeting.
Yep. Just to add another layer of complexity and potential, recent discoveries have unveiled RNA targeting CRISPR systems. The most well-known is Cas13.
Cas13, and it targets RNA, not DNA. Why is that a big deal? What's the advantage?
Targeting RNA opens up completely different possibilities because RNA is, generally speaking, transient. It's the working copy of the gene's instructions used to make proteins. DNA is the permanent blueprint.
So changes to RNA wouldn't be permanent?
Exactly. If you modify or degrade an RNA molecule using Cas13, the change isn't permanent in the cell's lineage. It's not passed down through cell division or to offspring. This makes RNA targeting potentially safer or more suitable for temporary effects.
Like what?
Like temporary gene knockdown, reducing the amount of a protein for a short time, or developing antiviral therapies by targeting and destroying viral RNA directly. Crucially, Cas13 systems have also been adapted for highly sensitive detection of nucleic acids.
Detection, like diagnostics.
Yes. Imagine rapid, accurate, maybe even cheap tests for detecting infectious diseases by spotting viral RNA or picking up cancer biomarkers or specific genetic markers. It could revolutionize diagnostics.
So CRISPR isn't just editing, it's detection too. This toolkit just keeps growing.
It really does. And it's important to note, while a lot of the ethical debate focuses on permanent DNA changes from standard Cas9, even these non-cutting tools like DCas9 platforms, base editors, and RNA targeting systems raise similar moral questions, depending on what they're used for and how long their effects last.
The constant evolution is just staggering. The capabilities are expanding so fast.
Which means the moral and practical stakes just keep getting higher, right?
Absolutely, because what we're really talking about here is reshaping our entire world. Medicine, agriculture, maybe even evolution itself.
This is where the implications get really tangible, moving from the microscope slide to, well, profound societal impact.
Okay, let's shift gears then. Let's move from the incredible science to the profoundly human impact. Because here's where CRISPR really hits home, right? Imagine. What if we could simply fix the faulty genes that cause inherited diseases? Permanently change the course of someone's health?
That is the central question, isn't it? The one driving this whole new era in medicine. A question that felt like pure science fiction just, what, a decade or so ago.
Yeah. Feels like yesterday.
The potential to actually cure genetic diseases is just immense, especially for those conditions caused by a single faulty gene, a single typo in the DNA.
Like, what sort of conditions are we talking about?
We're talking about devastating diseases like sickle cell anemia, that painful blood disorder affecting millions, mostly of African descent, or beta thalassemia, another severe blood disorder needing constant transfusions, cystic fibrosis impacting the lungs, the digestive system, even Huntington's disease, that awful progressive neurodegenerative disorder.
All caused by single gene mutations.
Primarily, yes. And CRISPR offers the potential to go in and correct that single mutation, offering real hope, maybe even a cure, where previously there was only symptom management or nothing.
And this isn't just theoretical hope anymore, is it? We're seeing this move into the clinic.
Exactly. We're not just talking lab bench science. In 2019, that was a monumental step. The first CRISPR-based therapy, called CTX001, was approved for clinical trials.
CTX001, what does it target?
It aims to cure both sickle cell disease and beta thalassemia. The approach is clever. They take the patient's own blood stem cells out of their body,
Okay.
edit them, using CRISPR in the lab, to fix the hemoglobin gene, basically getting them to produce healthy hemoglobin again.
And then put the corrected cells back in.
And then reinfuse those edited healthy stem cells back into the patient. And the early results, they've been incredibly promising. Some patients are showing complete remission of symptoms.
Complete remission, meaning they're cured.
It looks that way for some early participants. They no longer experience the debilitating pain crises of sickle cell or need the constant transfusions for thalassemia. It's a huge leap, a potential one-time cure for these lifelong debilitating conditions.
That's incredible. And even before that specific trial, CRISPR was being tested in people.
Yes, things moved very fast, particularly in some regions. Reports indicate that as early as May 2018, even before CTX001 started, at least 86 individuals in China had already received some form of CRISPR gene alteration as part of clinical trials.
For what kinds of conditions?
A whole range, actually. Multiple myeloma, various other cancers, even HIV-1 infection. It really underscores how quickly the clinical translation happened, though perhaps with varying levels of oversight and reporting in different places.
It really highlights the speed. But beyond these inherited blood disorders, what else is CRISPR being aimed at in medicine?
Oh, the potential is much broader. Cancer is a huge focus.
How can CRISPR fight cancer?
Several ways. One exciting area is engineering a patient's own immune cells, like T cells, to make them better at recognizing and destroying cancer cells, kind of turbocharging the immune system.
Immunotherapy, but enhanced by CRISPR.
Exactly. Or you could potentially use CRISPR to directly target and disable genes that are essential for the cancer cells to survive and grow, or even correct mutations that drive cancer growth in the first place.
And what about infectious diseases like HIV?
There's groundbreaking research there, too, trying to use CRISPR to achieve a functional cure for HIV. One approach is to target the CCR5 gene in a patient's immune cells.
CCR5, that's the gene Heat Jiankui targeted, right?
The very same. CCR5 makes a protein that HIV uses as a doorway to get into T cells. So the idea is, if you use CRISPR to disable the CCR5 gene in a patient's cells, you could make their cells resistant to HIV infection.
Wow. The ability to just precisely target and modify genes like this, it opens up therapies we couldn't have dreamed of before, really tackling the root cause of disease.
It's moving us closer to truly personalized medicine.
And it's not just human health, is it? You mentioned agriculture earlier. CRISPR is revolutionizing that field too, and also just basic scientific research.
Absolutely. The impact on agriculture and research is just as profound, potentially even broader in some ways.
Let's talk food security. It's a massive global challenge. The UN numbers are stark, nearly 800 million people undernourished, 2 billion lacking key nutrients.
And traditional crop breeding, while it's given us huge gains, is slow. It can take decades to develop and introduce a new desirable trait into a crop variety. Plus, it's often imprecise. You cross two plants, you get a mix of genes, some good, some maybe not so good.
Lots of trial and error.
A lot. CRISPR changes the game. It allows for incredibly precise genetic changes in potentially just a single generation. It's like using a surgical scalpel instead of a shotgun to modify the plant's blueprint.
What are some concrete examples? What are we seeing already?
We're already seeing crops engineered for better resilience and nutrition. Imagine entire fields of wheat that are resistant to powdery mildew. That's a devastating fungal disease. Causes huge yield losses globally.
Okay, disease resistance. What else?
How about rice varieties designed to survive floods? Flooding is a massive problem for rice cultivation in many parts of Asia. CRISPR can help create varieties that tolerate being submerged for longer.
That could stabilize food supplies in vulnerable regions.
Exactly. Tomatoes are being enhanced for better nutrition, maybe higher vitamin content, or traits that improve shelf life, which reduces food waste.
Less waste is huge.
And think about safety. Researchers have developed rice varieties with lower levels of heavy metals like cadmium and arsenic. These metals can be naturally present in soil and water in some areas and accumulate in the rice grain. CRISPR can reduce that uptake.
Making staple foods safer.
And then there's pest resistance. Crops engineered to resist insects could drastically cut down the need for chemical pesticides. That means more sustainable farming practices, healthier food for us, and less damage to the environment. Maybe reducing fertilizer needs too.
The potential impact on sustainable agriculture seems enormous. And what about livestock?
Yes, less discussed perhaps, but the potential is there too. Fortifying livestock, improving disease resistance in chickens or pigs, maybe increasing lean muscle mass in cattle for more efficient meat production, or even adapting animals to warmer climates.
It really paints a picture of a more resilient, healthier, more abundant food future, building resistance to climate change right into our food sources.
That's the goal.
And beyond food, you said CRISPR accelerates scientific discovery itself.
Profoundly. One of the biggest impacts is on creating animal models for research.
Like lab mice.
Exactly. Before CRISPR, making a mouse with a specific genetic mutation to mimic a human disease, say cystic fibrosis or Alzheimer's, could take months, sometimes years. It was laborious and expensive.
No.
With CRISPR, scientists can create these precise genetic models in weeks. It's dramatically faster and cheaper. This hugely accelerates research into understanding human diseases and, crucially, testing potential drugs and therapies.
So it speeds up the entire pipeline of medical discovery.
Massively. And it's not just mice. It's allowing researchers to study organisms that were previously very difficult to manipulate genetically. It's also enabled powerful new techniques like high-throughput screens.
High throughput, meaning testing lots of things at once.
Exactly. You can use CRISPR to systematically knock out or modify thousands of different genes across millions of cells simultaneously, then see what effect that has on, say, drug resistance or a particular cellular process. It helps rapidly map out entire genomic pathways involved in health and disease.
Pinpointing exactly which genes do what.
And how drugs interact with them. It's giving us insights into complex biology, like how fish fins evolved into limbs or how diseases develop at a speed and scale we've never seen before.
Okay, this leads us to something really, well, provocative. A big what-if question. Population-level control. What if we could tackle deadly diseases not just by treating sick people, but by altering entire species?
Ah, yes. Gene drive technology. Powerful and definitely unsettling for some.
It sounds like something straight out of science fiction. How does it even work? How can you change a whole species?
Okay, so gene drives are basically a genetic engineering trick. They use CRISPR, typically, to make sure a specific gene or trait spreads through a population much, much faster than it normally would through standard inheritance.
Faster than 50 inheritance.
Way faster. Normally, yeah, an organism has 2 copies of each chromosome and passes on 1 copy to its offspring. So a particular gene variant has a 50% chance of being inherited. A gene drive bypasses that.
What?
It includes the CRISPR machinery along with the gene you want to spread. In the offspring cells, the CRISPR system finds the other chromosome, the one that doesn't have the gene drive yet, cuts it, and then tricks the cell's repair system into copying the gene drive sequence over into the cut site.
Whoa. So it converts the other chromosome to match.
Essentially, yes. It ensures that almost all offspring inherit the gene drive, not just 50%. This means if you release just a few individuals carrying a gene drive into a wild population.
It spreads like wildfire through the generations.
Potentially, yes.
Yeah.
That specific gene can rapidly become universal, or nearly universal, throughout the entire species in a relatively short number of generations.
Okay, that's powerful. What are the potential public health applications? You mentioned disease control.
The potential benefits could be immense, truly revolutionary. Think about vector-borne diseases, diseases spread by insects or other animals. Malaria is the classic example, still kills hundreds of thousands every year.
Spread by mosquitoes.
Right. What if you could use a gene drive to spread a gene through mosquito populations that makes them resistant to the malaria parasite? so they just can't transmit it anymore.
You could wipe out malaria transmission.
That's the hope. Same idea for dengue fever, spread by different mosquitoes, or Chagas disease, Lyme disease, spread by ticks, schistosomiasis, spread by snails. If you can modify the vector organism using a gene drive so it can no longer carry or transmit the pathogen.
You could potentially eradicate these diseases, or at least control them on a massive scale.
That's the potential. Imagine a world where these ancient scourges are significantly diminished or gone. It's a huge motivator for the research.
Beyond disease, what about ecosystem management? You mentioned invasive species.
Yes, it offers a potentially powerful new tool there too, though also raises ecological concerns. For instance, you could potentially design a gene drive to reduce the fertility of an invasive species, like rodents on islands where they're wiping out native birds.
So control their population size genetically.
Or even eliminate them locally, potentially. Same idea for invasive plants that choke out native ecosystems. It could also be used, theoretically, to manage wildlife disease reservoirs, like reducing Lyme disease in mice, which then reduces transmission to ticks and humans.
But the potential for unintended consequences must be huge, right? If this spreads through an entire species, what if it jumps to another species? What if it disrupts the ecosystem in ways we didn't predict?
Absolutely critical concerns. And researchers are very aware of this. That's why a lot of effort is going into building safety features like off switches.
Off switches, how would that work?
Designing secondary gene drives that could be released to reverse the effects of the first one, or maybe building drives that are geographically limited or that self-destruct after a certain number of generations. The idea is to have ways to control, inhibit, or even eliminate the gene drive from the population if something goes wrong or if it's no longer needed.
Building in breaks from the start.
Essential. Because the power here is immense, the potential for irreversible change is real. Responsible development demands these kinds of safety considerations are built in from day one.
So, as we've seen, the applications are just nearly limitless, from fixing a single letter in DNA in one person to changing entire ecosystems. It touches every aspect of life.
But with that immense power comes equally immense responsibility. And that leads us straight into some of the most profound ethical questions humanity has ever had to grapple with.
We're really standing at the edge of possibility here, aren't we? Wrestling with questions that could reshape our definition of what it means to be human.
Exactly. This is where the cutting edge of science meets our deepest, most complex societal values, where the promise of incredible benefit has to be weighed against equally incredible caution.
Okay, let's dive into that ethical minefield. The core dilemma that jumps out immediately: therapy versus enhancement. Is it okay to use CRISPR just to treat or cure a recognized disease? Or is it permissible to go further, to enhance human features beyond treating illness?
Ah, the slippery slope.
Right, we're talking about things like, should we use it to make people taller, stronger, give them better vision than 20/20, or even boost cognitive factors like learning ability, memory? Where do you draw the line?
It is an incredibly difficult line to draw, maybe even an impossible one in some cases, because it's not always clear-cut. The main problem is defining what actually counts as a medical pathology that needs treatment versus what's just a variation within the normal human range, or even just slightly outside it.
Edges are fuzzy.
Extremely fuzzy. That makes the boundary between therapy and enhancement really murky. Think about it. Take cholesterol. If we use gene editing to lower someone's bad cholesterol levels, significantly reducing their risk of heart disease, is that therapy?
Seems like therapy to me, preventing disease.
Okay. But what if it makes their cardiovascular system so healthy that they live significantly longer and have more vitality than the average person? Is it still therapy then? Or has it crossed into enhancement? What if we could edit genes to guarantee someone lives to 120 in perfect health? Where did therapy stop and enhancement begin?
That's tough. Who gets to define what normal health even means, especially when we have the power to push way beyond current norms?
Exactly. And who has the authority to make these decisions? Not just for individuals, but for society. Should using CRISPR for medical enhancement be considered, say, a form of free speech or personal expression, a right to control your own biology?
Like bodily autonomy, but extending to genetics?
Kind of. And if it is seen as a right, how, if at all, should that right be limited? To prevent maybe exacerbating social inequalities or creating new forms of discrimination? Should governments regulate this, international bodies, or should it be purely an individual choice?
These are fundamental questions about liberty, equality, the role of medicine, what human flourishing even looks like.
Deep, deep questions. But perhaps the single greatest, most intensely discussed controversy around CRISPR involves heritable germline editing.
Germline editing, so editing DNA in eggs, sperm, or embryos.
Correct. Editing in a way that the changes will be passed down not just to the individual created from that embryo, but to all of their descendants forever.
So it's not just editing one person's genes, it's editing the human gene pool itself.
That's why it's the apex of the controversy. It involves not only the person directly treated, but potentially countless future generations who never consented. The stakes are incredibly high.
What's the general feeling about this globally? Is there any consensus?
Well, there's generally significant public support for using CRISPR therapeutically, in somatic cells, regular body cells to treat diseases in existing individuals. But germline editing, that's where you see widespread ethical and safety concerns across the globe.
So most people are against it.
Most professional scientific and medical societies currently state that heritable germline editing should be prohibited or at least paused, pending much more research and societal debate. However, it's not entirely black and white. Reports and recommendations from bodies in several countries, the Netherlands, the UK, Spain, and notably the U.S. National Academies of Sciences, Engineering, and Medicine, NASEM, suggest that maybe, just maybe, such editing could be permissible if, and only if, certain extremely stringent conditions were met.
So, not a hard no forever, but a no unless. Under what kind of conditions?
Extraordinarily stringent ones. The NASEM report, for example, laid out a whole list of prerequisites that would need to be satisfied before human heritable germline editing research could even begin. They really emphasize the profound moral weight here.
What are some of those conditions?
Okay, first, the absolute absence of reasonable alternatives, meaning if there's any other way to prevent the disease for the family, like pre-implantation genetic diagnosis, PGD, germline editing shouldn't be used. Second, strict restriction to preventing a serious disease or condition, absolutely no enhancement.
Okay, only for serious diseases with no other options.
Third, restriction to editing genes that are convincingly demonstrated to cause or strongly predispose to that disease, no speculative editing. Fourth, restriction to converting those genes to versions already common in the population and known to be associated with ordinary health, not creating new, supposedly superior gene versions.
So only restoring known healthy versions.
Right. Fifth, availability of credible preclinical and clinical data on the specific risks and potential benefits of the proposed edit. You need strong evidence it's safe and likely to work. Sixth, ongoing rigorous oversight during clinical trials, including plans for long-term multi-generational follow-up while still respecting the autonomy of participants and their descendants.
Multi-generational follow-up, wow.
Seventh, maximum transparency, consistent with patient privacy. Eighth, continued reassessment of the health and societal benefits and risks, with broad public participation. This can't just be scientists deciding. And ninth, crucially, reliable oversight mechanisms to prevent any extension to non-disease uses, to start that slippery slope to enhancement.
Those are incredibly high hurdles. It sounds like they're saying maybe, but only in the rarest of rare circumstances and with extreme caution and oversight.
That's exactly the message. It reflects the immense responsibility of altering the human germline.
But even with those hurdles, there are arguments in favor of germline editing, right? Why would someone advocate for it?
Yes, there are counter-arguments. Some argue there's a moral imperative to protect future children from inheriting devastating genetic diseases if we have the means, the protection of defective embryos argument. Others suggest some diseases might be best tackled at the earliest possible stage in the embryo for the most complete cure. And then there's the argument, again, about reproductive freedom or free expression. Should parents have the right to choose this technology for their future children, especially if it prevents severe suffering?
It's such a complex ethical balance, deeply held values on both sides. And then right in the middle of this debate came the Hejiankui incident.
Indeed. The Hejiankui case in 2018 was a bombshell. It served as a stark global wake-up call.
Remind us what happened.
He, a Chinese scientist, announced at an international conference that he had used CRISPR to edit the genes of twin girls, Lulu and Nana, when they were embryos. His goal was supposedly to make them resistant to HIV infection by disabling that CCR5 gene we mentioned earlier.
And he just announced it. Had he gone through ethical reviews?
Apparently not adequately. The announcement was made without broad scientific consensus that it was safe or ethically permissible, and seemingly bypassed standard ethical review processes. It caused immediate shock and widespread condemnation from the scientific community worldwide. People were horrified.
What happened to him and the twins?
Jiang Kui was ultimately sentenced to prison in China for violating regulations and conducting illegal medical practices. The health and long-term effects on the twin girls remain largely unknown, which is another layer of ethical concern.
That case just vividly illustrates the potential for misuse, doesn't it? Or at least for reckless, premature application before we really understand the consequences or have societal agreement.
Absolutely. It highlighted the urgent need for clear, globally agreed-upon guidelines and regulations. It showed that just because the technology exists doesn't mean it should be used in humans yet, especially not on the germline, without extreme caution and broad societal consensus.
And this fear of misuse, or even well-intentioned overreach, inevitably brings up the horrifying specter of eugenics.
Yes, that historical parallel casts a very long, very dark shadow over any discussion of germline editing. There's a real profound societal risk that allowing CRISPR germline editing, even if intended strictly for medical purposes, could inadvertently open the door to a return of eugenics.
Remind us what eugenics is, fundamentally.
At its core, eugenics is the belief that the human population can somehow be improved by controlling heredity, by encouraging reproduction among those deemed to have desirable traits and discouraging or preventing reproduction among those deemed to have undesirable traits.
Selective breeding for humans.
Essentially, yes. Historically, this involved actively weeding out people with so-called bad genes through forced sterilization, discriminatory immigration laws, institutionalization, and promoting the breeding of those with good genes. And it led to horrific atrocities.
It wasn't just fringe extremists, was it?
Tragically, no. These practices were often backed by state authorities, mainstream scientists, doctors, and educated elites who genuinely believe they are acting progressively, scientifically, for the betterment of society.
Yeah.
That's what makes it so chilling.
Is there a specific historical example that really drives this danger home?
The 1927 United States Supreme Court case, Buck v. Bell, is a truly horrifying one. The highest court in the U.S. upheld a Virginia law that allowed the state to forcibly sterilize individuals considered mentally unfit.
Forcibly sterilize them?
Yes. The case involved Carrie Buck, a young, economically disadvantaged woman. She was labeled feeble-minded, supposedly like her mother and her own infant daughter had been deemed likely to be. Despite evidence suggesting she was actually of normal intelligence, the state ordered her sterilized.
And the Supreme Court said that was okay?
They did. Justice Oliver Wendell Holmes, Jr., himself a proponent of eugenics, wrote the infamous majority opinion, which included the line, "3 generations of imbeciles are enough." It was a shocking justification for violating your fundamental rights.
What was the impact of that ruling?
It led to the compulsory sterilization of over 50,000 Americans, disproportionately targeting the poor, minorities, and those with disabilities. It also provided a legal precedent that, horrifyingly, influenced the Nazi regime's racial hygiene programs in Germany.
And that ruling, it's still on the books.
Unbelievably, yes. Buck v. Bell has never been explicitly overturned by the Supreme Court. It stands as this stark, concrete historical example of how easily society, even a democracy, can commit egregious systematic errors when it tries to control human genetics based on flawed science and societal biases. It's a powerful warning about the potential for good intentions to pave the road to terrible outcomes.
It really highlights the danger when scientific capability outpaces ethical wisdom and societal safeguards.
Precisely. And this historical context reinforces the deep concerns about the inherent uncertainty and unpredictability involved in gene editing, especially germline editing.
Even if the edit itself looks perfect at the DNA level.
Yes. Even if you make the exact DNA sequence change you intended, the downstream biological consequences, particularly for complex traits, can be unclear and unpredictable.
Why is that?
Because most biological traits, think height, intelligence, risk for common diseases like diabetes or heart disease, aren't determined by just one gene acting in isolation. They result from the incredibly complex interplay of numerous genes, regulatory elements like enhancers, environmental factors throughout life, and epigenetic modifications. It's a symphony, not a solo instrument.
We just don't understand the whole symphony yet.
Not even close. This profound complexity means that even with a perfect edit to one gene, we might inadvertently disrupt other pathways or create unforeseen problems down the line, maybe even generations later. This fundamental uncertainty makes doing an accurate risk-benefit analysis incredibly difficult, which obviously complicates the moral decision-making.
We could be causing new problems while trying to fix old ones.
That's the fear. And it's not just the long-term biological uncertainty either. There are still inherent technical limitations with the CRISPR technology itself that remain significant concerns, especially for widespread clinical use.
What kind of technical limitations?
Well, one is limited on-target editing efficiency. Sometimes the CRISPR system just doesn't make the intended edit successfully in all the target cells. You might only get 50% or 80% efficiency.
So not a complete fix.
Right. Another issue is incomplete editing, or mosaicism. This is particularly relevant for editing embryos. You might end up with an individual where some of their cells have the edit and others don't. A patchwork. The health consequences of that can be really unpredictable.
A mix of edited and unedited cells in the same person. Yeah, that sounds complicated.
And then the big one, inaccurate editing. This includes off-target effects, where the CRISPR system cuts DNA at unintended locations elsewhere in the genome, places that just happen to look similar to the target site.
Cutting in the wrong place.
Exactly. Or even on-target errors, where it cuts at the right place, but the cell's repair process makes mistakes, leading to larger insertions, deletions, or rearrangements right at the target site.
These unintended cuts or rearrangements could cause new problems, like new diseases, or even cancer.
That is the concern, yes. Unintended genetic damage could potentially disrupt important genes or trigger cancer development. Now, the technology is constantly improving. Scientists are developing higher fidelity Cas9 versions, better guide RNA designs, more sophisticated delivery methods to minimize these risks, but they haven't been eliminated entirely. These technical limitations remain principal worries about widespread use, especially for something as irreversible as germline editing.
So the ethical concerns aren't just abstract philosophy. They're deeply tied into the practicalities, the technical limits, the biological unknowns, and also who actually owns and controls this incredibly powerful technology.
Exactly. These practicalities are inseparable from the ethics. Which brings us to the whole battle for the blueprint itself, the patents, the access, the global governance.
And what a battle that has been, right? The moment CRISPR-Cas9's potential became clear, the race for intellectual property was on. It started with those foundational patents from 2012 by Emmanuelle Charpentier and Jennifer Doudna.
That really marked the turning point, not just scientifically, but commercially. It set the stage for some major IP disputes.
Who are the main players holding those key initial patents?
The primary owners of those foundational patents are collectively known as the CVC Group. That includes Emmanuelle Charpentier herself, the regents of the University of California, where Doudna works, and the University of Vienna, where Charpentier was affiliated.
CVC Group. And what does their patent portfolio cover? Is it just the basic idea?
Oh, it's quite broad. They were smart about it. Their portfolio covers the core compositions.
Yeah.
Now, Cas9 protein plus the guide RNA, either as two separate molecules, dual guide, or combined into one, single guide RNA. It covers methods of using these compositions to cut DNA. It covers different ways of delivering them into a cell. And importantly, it also covers the use of those mutated Cas9 variants we talked about earlier.
Like the nickase N-Cas9 and the dead Cas9 or D-Cas9.
Exactly. Their patents cover using on Cas9 for making single-strand nicks and using D-Cas9 as that delivery platform, for example, bringing other proteins to specific DNA sites for gene regulation, like CRISPR interference, CRISPR-A, or CRISPR activation, CRISPR, and even for base editing. They cast a really wide net with those initial claims.
So they really tried to cover the foundational technology and its immediate variations. How is this IP actually managed and licensed out?
Well, ERS Genomics plays a big role. It was co-founded by Emmanuelle Charpentier in 2014. ERS acts as a global licensor for the CVC patent portfolio. They grant non-exclusive licenses across different sectors, research tools, diagnostics, agriculture, industrial biotech.
Non-exclusive, meaning multiple companies can get a license.
Generally, yes, for many applications. However, for certain high-value fields, there can be exclusive deals. For instance, CRISPR Therapeutics, which is one of the leading companies developing human therapies, holds the exclusive license from the CVC Group specifically for human gene and cell therapy applications.
Ah, so the rights get carved up depending on the field of use. That creates a complex landscape.
Incredibly complex. And the sheer scale of patenting activity around CRISPR is just mind-boggling. By 2020, something like over 12,000 CRISPR-related patent applications had been filed worldwide.
12,000?
Falling into around 4,600 distinct patent families. It's a true reflection of the intense commercial interest and ongoing innovation. Cas9 systems and newer systems like Cas12a are major areas of focus.
To navigate this, you mentioned distinguishing between fundamental and supplemental patents. How does that help understand the situation?
Right. That distinction is really important. Fundamental patents cover the core. enabling technology itself. Think of them like the land deed. If you want to build anything on that land, you need permission from the owner.
So any use of CRISPR requires a license to these fundamental patents.
Generally, yes. Historical examples would be the Cohen-Boyer patents back in the 70s for recombinant DNA. You couldn't really do genetic engineering without licensing them. Or the Cabelli patents for making monoclonal antibodies, vital for many drugs today. The CVC group's patents are largely considered fundamental for basic CRISPR-Cas9 use.
Okay, and supplemental patents.
Supplemental patents cover specific improvements, applications, or variations of the core technology. Maybe a new way to deliver CRISPR into cells, or modified Cas9 with better specificity, or using CRISPR for a very specific diagnostic test. These build on top of the fundamental technology.
So you might need a license for the fundamental patent and another license for the supplemental patent if you want to use that specific improvement.
Exactly. This creates layers of intellectual property. It can incentivize innovation by protecting specific improvements, but it can also create patent thickets that make it complicated and expensive for researchers or companies to actually develop and commercialize new products, because they might need licenses from multiple patent holders.
And this complexity was made even worse by different patent rules in different parts of the world, right? That fueled the patent wars.
Absolutely. A key difference was the U.S. system versus most of the rest of the world regarding when you had to file. Most countries operate on a first-to-file system. Pretty simple. The first group to get their patent application describing the invention into the patent office wins the patent. China, Japan, Europe all work this way.
First come, first served at the patent office.
Basically. And the U.S. switched to first-to-file as well, but only in mid-March 2013.
What was the U.S. system before March 2013?
Before that, the U.S. had a unique first-to-invent system. This meant it didn't matter who filed first. What mattered was who could prove they actually conceived of the invention and diligently worked on it first.
How could you prove that? Lab notebooks.
Exactly. Courts might have to examine lab notebooks, emails, witness statements, publication dates, anything to establish the timeline of invention. This system often led to complex legal battles called interference proceedings to determine who was truly first.
And the timing of this U.S. rule change was critical for CRISPR.
Incredibly critical. The CVC group filed their key provisional patent applications under the old first-to-invent rules just before the deadline. The Broad Institute filed slightly later, some under the old rules, some under the new first-to-file rules. This difference in timing and applicable rules played a huge role in the initial disputes, especially in the U.S.
So who was actually first in the race to the patent office? You mentioned a Vilnius group.
Right, there was a group based in Vilnius, Lithuania, led by Virginijus Kukenis. They were technically the first to file a couple of provisional applications describing a CRISPR system. However, a critical point was that their early applications apparently didn't explicitly describe the necessity of the track RNA component for the system to work efficiently in the way Doudna and Charpentier later showed. That detail proved crucial.
Okay, so Vilnius was early, but maybe missed a key piece. What about CVC?
The CVC group, Charpentier and Doudna, filed their first provisional application about 2 months after Vilnius, but their filing was incredibly detailed. It explicitly described the 3 essential components: the CRISPR RNA, cRNA, the transactivating cRNA, triad RNA, and the Cas9 protein.
They identified all the key players.
Yes, and crucially, they also described how the cRNA and tracRNA could be fused together into a single artificial single-guide RNA, sgRNA. That simplification is what most labs use today.
Did their first filing show it working in human cells?
Their very first provisional application didn't include experimental data showing it working in eukaryotic cells, like human, plant, or animal cells. However, it did describe methods for how to do that. And shortly after filing, in June 2012, they published their landmark paper in Science demonstrating the system worked beautifully in vitro in test tubes.
Okay, then came the Broad Institute.
Right. The Broad Institute, associated with MIT and Harvard, led by Feng Zhang, filed their first provisional application about 4 months after CVC. A key difference was that the Broad's filing did include some initial experimental data showing CRISPR-Cas9 working in eukaryotic cells.
Ah, so they had the eukaryotic data first in a patent filing.
In a patent filing, yes, it seems so.
Hmm.
Although CVC described how to do it earlier. Then other groups like Tulgin from South Korea and Sigma Aldrich filed even later, 6 months or more after CVC, also presenting some eukaryotic data. Tulgin tried to differentiate itself by focusing on using CRISPR for targeted gene insertion.
But you mentioned earlier that multiple labs quickly replicated the eukaryotic work after CVC's publication.
Exactly. That's a crucial point. Within just 6 months of CVC's science paper being published, which clearly laid out the components and mechanism, 5 independent labs around the world published papers showing they had successfully gotten CRISPR to work in eukaryotic cells, following CVC's lead. This really demonstrated the clarity and enabling nature of CVC's original work.
So fast forward through years of legal battles, where did things land in the U.S., say, as of 2022 or 2023?
It's still somewhat complex in the U.S. In February 2022, the U.S. Patent Trial and Appeal Board, PTAB, made a significant ruling in one of those interference proceedings. They essentially ruled in favor of the Broad Institute, finding that their claims regarding the use of single-guide CRISPR-Cas9 systems, specifically in eukaryotic cells, were patentably distinct from CVC's earlier invention, which is initially demonstrated in vitro.
So Broad got the patent rights for use in human cells.
For single-guide systems in eukaryotes, yes, according to that specific PTAB decision. However, and this is important, the PTAB confirmed this decision did not invalidate or impact any of CVC's many other granted U.S. patents. CVC holds over 50 U.S. patents by now, covering broader aspects like the compositions themselves, the use of CRISPR in a cell generally, without being limited to eukaryotes, and the use of single-guide RNA itself.
So both groups have valid U.S. patents related to using CRISPR in human cells.
That seems to be the current situation, yes. The Broad and CDC patents effectively coexist as they relate to eukaryotic use. If you want to commercialize a CRISPR therapy in the U.S., you likely need licenses from both CVC, or its licensees like CRISPR Therapeutics, and the Broad Institute.
And the legal fight isn't even over.
Nope. Both CVC and Broad appealed that 2022 PTAB decision to the federal courts. The legal briefing was still ongoing in 2023. So the final chapter in the U.S. patent saga may not be written yet.
What about Europe? Has it played out differently there?
Yes, the European patent landscape looks quite different. Generally speaking, CVC has maintained a much more dominant IP position in Europe. Several key patents held by the Broad Institute, Sigma, and Toolgen related to CRISPR have actually been revoked by the European Patent Office, EPO.
Revoked?
Often due to specific aspects of European patent law, which can be very strict. For example, one of Broad's foundational patents covering CRISPR use in eukaryotes was revoked due to a technical flaw in how they claimed priority back to their earlier U.S. applications. Essentially, a paperwork error invalidated their claim in Europe.
A paperwork error cost them a foundational patent. Wow.
European patent law can be unforgiving on procedural matters. Similarly, a Sigma patent was found invalid for lacking an inventive step, obviousness, and a Toolgen patent was revoked on procedural grounds related to late amendments.
So CVC's patents are generally upheld in Europe?
Yes, several of CVC's foundational European patents have survived opposition challenges and been upheld. These cover the core single-guide system, its use in cells generally, not limited to eukaryotes, and also dual-guide systems in eukaryotic applications specifically. While one CVC patent was revoked based on strict written description requirements, they preserved the subject matter in other filings. So overall, CVC is in a much stronger position in Europe compared to the contested landscape in the U.S.
These huge, complex, expensive patent battles, they must have implications for who actually gets to use the technology and how much it costs, right? Which brings us back to that critical issue of equitable access.
Absolutely. The innovation is amazing, but it raises serious concerns about fairness, about distributive justice, who benefits, who gets left behind.
Because these new CRISPR therapies, they're not going to be cheap, are they? Especially initially.
Almost certainly not. Like most cutting-edge biomedical advances, they're expected to be highly profitable for the companies and patent holders involved. The initial prices for CRISPR-based gene therapies are likely to be extremely costly, potentially hundreds of thousands, maybe even millions of dollars per treatment.
Millions per treatment. That immediately raises ethical flags, especially considering the public funding involved in the basic research.
Exactly. A huge amount of the foundational research that led to CRISPR, the basic science understanding the bacterial immune system, was funded by government grants, by taxpayers. So, is it fair, is it just, to then potentially deny the life-saving benefits of that publicly funded research to the very taxpayers who paid for it, simply because the final product has an unaffordable price tag?
It feels deeply problematic.
It does. It creates situations where individuals or families facing devastating diseases who could potentially be cured by CRISPR might be forced into impossible choices, choosing between a life-saving therapy and bankruptcy, or foregoing treatment altogether because they can't afford it. The potential for severe economic hardship, on top of the physical and psychological burden of illness, is immense.
And this isn't just a problem within wealthy countries, right? What about global equity? How do these potentially life-changing therapies or even CRISPR-enhanced crops reach people in the most impoverished parts of the world?
That's an even greater challenge. How do you ensure that CRISPR-modified foods, designed for better nutrition or drought resistance, actually reach and benefit smallholder farmers or undernourished populations in low-income countries, especially in regions plagued by conflict or political instability?
The market alone won't solve that.
Probably not. How do you incentivize companies whose primary driver is profit to develop and distribute products for populations that have very little purchasing power? It requires thinking beyond standard commercial models, maybe exploring things like tiered pricing, patent pools for humanitarian use, public-private partnerships, or, as some suggest, stronger regulations like anti-price gouging laws to prevent exploitation. But it's a massive global challenge.
This really underscores the need for some kind of oversight, doesn't it? Not just on the ethics of using CRISPR, but on how it's developed, patented, priced, and distributed.
Absolutely. Which brings us to the call for international oversight and governance.
Because ethics statements and guidelines from scientific societies, while important, they don't have legal teeth, do they?
Exactly. They are crucial for shaping norms and guiding responsible conduct within the scientific community. But violating them usually leads to relatively minor consequences, maybe losing grant funding, having a paper retracted, perhaps losing your job. It's not the same as breaking a law, which can result in heavy fines or even prison time, as we saw with He Jiankui.
So we need more than just guidelines. We need actual regulations, maybe even international treaties.
Given the immense potential of CRISPR for good, but also the dark history of eugenics, the very real risks of germline editing, and the theoretical potential for misuse, whether for bioweapons or creating genetic casts, it seems entirely reasonable, even necessary, for the global community to establish clear national and supranational regulations.
Updating existing frameworks.
Possibly. It might even require revisiting and revising older international agreements, like the Geneva Conventions, related to warfare, or the Biological and Toxin Weapons Convention, to explicitly address the new capabilities offered by powerful genomic engineering technologies like CRISPR. We need to ensure they can't be weaponized or misused in ways the original framers couldn't have imagined.
It's not just about guiding science anymore. It's about governing a fundamental new human capability.
That's a perfect way to put it. It transcends national borders, so the governance needs to be collaborative and international.
Are there promising ideas or initiatives emerging for how to actually do this kind of collaborative global governance?
Yes, there are some interesting developments. Within the scientific community, for example, the Association for Responsible Research and Innovation in Genome Editing, ARIGE, was founded by European scientists specifically to foster dialogue, examine ethical issues, and provide guidance on genome editing practices globally.
So scientists trying to self-regulate responsibly?
To some extent, yes, and to inform broader discussions. But perhaps a more far-reaching idea comes from scholars like Sheila Jasanoff and Benjamin Hurlbut. They've proposed establishing an international, interdisciplinary, global observatory for gene editing.
A global observatory? What would that entail?
Their key argument is that deliberations about the profound moral and societal issues raised by gene editing should not be dominated solely by scientists or scientific bodies. Given the huge societal implications, the discussion needs to be much broader.
Involving ethicists, policymakers, the public.
Exactly. They envision a broad network similar to international networks focused on human rights or climate change. It would bring together scholars from diverse fields, ethics, law, social sciences, humanities, along with policymakers, civil society organizations, and public representatives from different cultures and nations. The goal is to foster deep, ongoing exchange and deliberation across all these divides.
That makes so much sense. It can't just be the scientists deciding for everyone else. It needs to be a truly global conversation reflecting diverse human values.
Precisely. Because as the technology inevitably continues to evolve at this breakneck pace, our discussions about the ethical and legal frameworks have to evolve alongside it. It's not a one-time conversation.
So who enforces whatever rules or guidelines emerge?
Ultimately, the enforcement of research laws and ethical guidelines will likely fall to a combination of actors: national governments through their legal and regulatory systems, principal investigators ensuring ethical conduct within their own labs, and institutional review boards, IRBs, or ethics committees that oversee research proposals at universities and research institutions. It will be a multi-layered system of governance.
An ongoing, evolving, and critically important process for our future.
Absolutely. For the foreseeable future, yes.
We have covered so much ground today. We've unpacked CRISPR, this incredible tool born from, of all things, ancient bacterial defenses against viruses, and now it's capable of literally rewriting life itself.
It's mind-boggling when you think about its origins.
From potentially curing devastating genetic diseases like sickle cell, to fortifying our crops against climate change and pests, maybe even battling global pandemics with new diagnostics or therapies, or shaping entire ecosystems with gene drives. The applications are just vast and awe-inspiring.
The potential for good is truly immense.
Yet, as we've really dug into, we've also confronted these profound ethical tightropes we're now walking, that blurry, difficult line between therapy and enhancement, the chilling specter of eugenics learning from the horrific mistakes of the past, like Buck v. Bell.
And the very real concerns about unintended consequences, both biological and societal.
Plus the complex, high-stakes battles over patents and intellectual property, and the critical challenge of ensuring equitable access, making sure this powerful technology doesn't just benefit the wealthy.
The story of CRISPR, it's clearly not just another scientific breakthrough. It feels different. It feels like a profound test for humanity itself.
A test? How so?
Well, these molecular scissors have given us an unprecedented ability. The ability to play a direct role in evolution, a role previously unimaginable, to interact with and modify the very blueprint of life on this planet.
So the ultimate question isn't just what we can do with this incredible power.
Exactly. The deeper question is how wisely, how ethically, how justly we choose to wield it. Knowing, really knowing, that the decisions we make today, the lines we draw, the regulations we put in place, they will literally echo through the generations. They will shape the future of life on Earth for centuries, maybe millennia, to come.